Good afternoon, everybody. My name is Caroline Lowe, and I'd like to welcome you to today's webinar. I'll be chairing the meeting, and I'm joined by Fred Hancock and Nessa Carlson, who will be helping field the questions, which we'll have at the end of the talk. This is a presentation which has been hosted by the Society of Chemical Industries Fine Chemicals Group and the Young Chemists panel. And our job is to facilitate links between organic chemists in industry and academia, and to provide a forum for discussion and sharing of information. And we've set up these webinars to support our colleagues through the COVID crisis when you're all working in such different situations. So there will be a question and answer session at the end of the presentation, and if you would like to answer a question, please type your question into the Q&A box at any time, and my colleagues will collate those, and we'll try and get as many of those answered as possible. We would like to extend a huge thank you to our corporate partner AstraZeneca, who are supporting the entire series, and sometimes you'll see that some of these will be supported by individual companies as well, but we'd like to thank everybody who's made this series possible. Can I also bring to your attention, we are running a national undergraduate online poster competition, and you can find the posters posted on this website here. They will be there till the end of the week, and I can encourage as many of you as possible to go and take a look, and you can comment on the posters and talk to the presenters. And just whilst I've got your attention, the next webinar will be from Kevin Lamb from the University of Greenwich, and he will be talking about electrochemistry. And the one after that will be from Justine Slavou from Johnson Matthey and Stephen Thomas from the University of Edinburgh, and they will be talking about biocatalysis and main group catalysis for hydroboration reactions. So I hope you will join us for some of those. Which brings me to introducing today's speaker, who is Lars Bärfacker from Bayer. He's currently actually on holiday in France, but he's joined us. This is a special appearance for today. Lars joined Bayer Healthcare in Wuppertal as a medicinal chemist in the year 2000. And since then, he has received a number of prestigious awards from Bayer, including the Global Drug Discovery Award in 2008 and the Otto-Bayer-Medal in 2014. In 2011, he moved to Berlin to work on oncology projects, and in 2014 he returned to Wuppertal, where he has been working on cross-functional aspects of drug discovery and drug delivery. And he's going to tell us today about the discovery of Finerenone, which is an MR antagonist for the treatment of cardiorenal diseases. And I would like to hand over the presentation to Lars.
So, first of all, I would like to thank the organizers for actually giving me the opportunity to talk about actually a project that keeps me busy now for more than 15 years. But I also would like to thank everybody who took the opportunity to log on for this presentation. To start this journey, of course, you need an indication which keeps you interested. And actually the beginning of the whole story was driven by our efforts to find a treatment for chronic heart failure conditions. And well, heart failure is actually a disease in which the heart is no longer to do its job. And basically the heart is nothing else than actually a pump that allows to push the blood around the body and to supply our organs with oxygen, basically. And there are, I would say, two conditions which are very important. One is the systolic version, which is actually the reduced pump function in terms of the ejection fraction of the heart. And the other one is the diastolic heart failure, which is more the inadequate filling on the other side of the pump, as you will. The prevalence in Western countries is, or worldwide, is very high. And looking at the last 3 decades throughout the aging population, the number of cases actually tripled. I have here some costs for Germany from the year 2008, but I'm pretty sure that Germany reflects the conditions we are facing in all westernized countries if you normalize it, let's say, on the population. The treatments available are basically lots of different drugs which are applied in parallel. However, this condition is still serious. So in the end, if the disease is progressing, you really need to come up with well, severe measures like pacemakers, or even in worst case, you need a transplantation, which actually sums up to mortality, which is in the range of 50% over 5 years. So what I want to talk about today is actually aldosterone antagonism, or more precise mineralocorticoid receptor antagonism. And this is actually a very, very old story, since the first anti-steroids that actually reached the market was actually Spironolactone, which were used for diuretic management of edematous conditions and essential hypertension. Then in the late 80s, somebody figured out that the MR receptor is also expressed in the heart. And this triggered actually a lot of efforts that were ending up in the setting up of the RAILS study, which was the first study to show that an anti-steroid can actually have significant benefits on primary endpoints in heart failure studies, like reduction of mortality and morbidity. And this actually opened up really a rally of a lot of companies to find novel MR antagonists that were able to address the limitations of spironolactone as a drug. I will come to that a little bit later. And since then, Finerenone was found as a second-generation MR antagonist, and it also showed that actually patients who are suffering from acute conditions of myocardial infarction are also benefiting from MR antagonist treatment. And Finerenone also in 2011 showed that milder conditions are also benefiting from this kind of a drug. So this seems more like a told story, but actually what are the difficulties, or let's say the improvements that are still to be made? So looking at our steroids that we are playing around with here, you have on one side aldosterone as, let's say, the bad guy who is doing some bad stuff in terms of hypertrophic and fibrotic conditions. On the other side, steroids, you have testosterone and progesterone, which are also, let's say, structurally similar, and also their receptors are a little bit relatives. So what is spironolactone actually doing? Spironolactone is blocking the binding of aldosterone to the mineralocorticoid receptor, and also is blocking the testosterone receptor. This can, of course, lead to severe side effects, and you can see there a picture of a man who is suffering from gynecomastia. This is actually a severe and painful growth of the male breast, and of course, this is a little bit more than, let's say, a small side effect. You can see that this is an effect on the left side here in the black box. It's dose dependent, And also in the rail study, it was shown to be an important factor. Another aspect is that spironolactone is not only blocking steroidal effectors, it's also having agonistic effects on the progesterone effectors. And this can lead also to the generation of impotence and menstrual irregularities. Also something you like to get rid of. And this actually led to the search for different and other treatment paradigms. And what I said here is pictured right now on the steroidal receptor profile. And you see you have a potent MR antagonistic activity with spironolactone, but it also affects the aldosterone, the testosterone receptor and the progesterone receptor. Spironolactone is a second generation antagonist, and you can see that on the EMR is significantly less potent. But the resulting or residual activities on the other steroidal receptors can be neglected. It's basically not active in this regard. I have mentioned gynecomastia, but what's important is with spironolactone, which does not show this kind of adverse event profile, that spironolactone, due to its lower potency on MR, is also having a lower clinical efficacy compared to spironolactone at the highest approved daily treatment dose. And this is not due to the limitation which comes from gynecomastia. There's another important side effect that is driven by an effect that we call hypokalemia. That is actually, hypokalemia is an effect that is raising systemic potassium levels that can lead to severe conditions of arrhythmia. And in this regard, you cannot dose up to a point where you have a better efficacy in the, let's say, on the effects you would like to achieve in the clinical study. So, as mentioned, a lot of companies entered the race for mineralocorticoid receptor antagonists, and actually Bayer also. And what we have been able to show is that dihydropyridines are maybe a good starting point, since they are having a significant effect on the MR. And you can see there's also some effects on the other receptors like GR or the androgen receptor or progesterone receptor. Everyone who is a little bit familiar with this kind of compound class will get to know, of course, that these have a longer history. And the history is also at Bayer shown as here. These compounds are very, very potent L-type calcium channel blockers. And of course, when we got to know that these are compounds that are active on the MR, we also tested the other compounds. And we have been able to show that also these compounds are low potency binders. So this was rather interesting for us. But not only for us. Here is our first patent applications. You will not be able to read it, but it's also not important. What is important is the priority date, which is middle of March 2004. And 3 weeks later, Takeda published also a patent application that was describing dihydropyridines as MR antagonists. Meanwhile, we are aware that also Novartis, Merck found these compound classes since these compounds are present in almost every kind of repository of any pharma company, and so this was not a surprise. I've heard from a colleague from Novartis that they would have never accepted a dihydropyridine as a starting point. Maybe this is a little bit now just illustrating the degree of desperation we had at that time, since we actually took the challenge and tried to bring these compounds to a level on which we can say, well, they have a good chance to make it as a drug. So what are you doing when you're actually facing a good hit? Basically, the first thing you do is, if you have a racemate in hand, try to separate it into the enantiomers and see whether you have a stereo-specific binding. And this is also what we did here. So you see on the left side the racemate, and in the middle actually the more active enantiomer. And you see that the potency is already quite okay-ish. However, the compound is almost equipotent towards the L-type calcium channel. That is, of course, something you do not want to have if you want to treat heart failure patients, because this, in the end, will cause a strong drop in blood pressure, and this is not what you actually would like to do in this kind of patients. The other enantiomer is significantly less active. This was already good sign. So basically, the first optimization parameter to solve was get rid of the L-type calcium potency, and of course, an intrinsic problem with this compound class is always half-life and metabolic stability, aside of some others. Here, I wrap up around 2 years of work in half a slide. Basically, we have been doing around 1,500 compounds to get a better understanding of SAR, and here I depicted just a couple of them as representatives. So on the upper left side, you find core replacement, and all the other core replacement we actually checked were basically inactive. On the below, you see that N-alkylation of the dihydropyridine core leads to virtually inactive compounds. Then we tried to improve metabolic stability by incorporating amlodipine kind of residue. You see in dark green the 2-amino etoxy kind of moiety, which is also illustrating that simply improving or incorporating solubilizing elements that did not work. Then in green, you see that at least some of the derivatives we have been synthesizing do actually work. So you can go from an ester towards a ketone, and basically you keep the level of activity. The first breakthrough actually came when we introduced Finerenone in the head group, which was actually raising the potency of the compound significantly. And at that point, we became interested, what is actually the potency driver? Is it more a directed kind of a bonding of the ketone towards, let's say, the protein backbone, or is it more lipophilic or a lipophilically driven potency gain that can be explained by the benzo group? And we tried a lot to get a better understanding, and when we came to the Chromenone showing on the left side, that was really the breakthrough in this regard, since we could show that you can keep the potency basically by introducing a different heterocyclic system which is completely lacking, yeah, the greasy part of the benzo group shown with a red arrow. When you actually made minor transformations by converting the nitrile into a ketone, then you basically were able to keep the potency that we actually started from with the compound on the upper red side. Then we conducted a small kind of panel of compounds to really check on the best, let's say, compound in terms of their overall profile, including, of course, PK. And with the ester on the upper part, which is even more potent, we did not achieve high bioavailability, probably based on first pass clearance with the compound. And actually, the best overall profile we actually managed to incorporate was with an ethyl ester that, as a racemate, was 53 nanomolar. So, as mentioned, in this direction, you always start and separate the enantiomers, and this is what we did, and we came up with a compound which is called BR4628, which is 28 nanomolar on the MR, and has actually a good selectivity profile, especially also compared towards the L-type calcium channel. And this was actually the first breakthrough, since always we were asked by our management, yeah, whether all the good data we produce in vivo and so on are really due to the MR activity, or whether it comes from L-type calcium channel modulation. And with this compound, we have been able to show that the MR is really the driver of efficacy. On the left, in the left box below the racemate, you see actually the in vitro profile showing 3 curves. Although you can't see the third one, this is due to the fact that spironolactone and BR4628 are actually almost co-spotting. So in this regard, BR4628 is really as potent as spironolactone. And this was actually a point where we decided to go into in vivo characterization. And we used a model that is not really, let's say, disease relevant. It's more proof of mechanism type of experiment. So what you can do is you can go into high aldosterone animals and try to counteract the high levels of aldosterone. You can actually then measure in the urinary, in the urine, the sodium and potassium levels. And since MR is actually modulating sodium retention, you will see, depending on the potency of MR, you will see an increase in sodium to potassium ratio. And as you can see here, comparing BR4628 with spironolactone, the compound is at least similarly active as spironolactone also in vivo. This was really a breakthrough for us, since we know, or knew now, that actually we can make it. So we tried to still improve the overall compound profile, maybe also get rid of the last level of selectivity problems we have in hand, and actually went to the next stage of optimization. To do that, we want to understand, of course, how the compound is actually binding. And at that time, actually, we started a collaboration with a group from Paris with Marie-Edith Raffarin-Aubelin, who actually made the first crystal of the upper structure of mineralocorticoid receptor. And based on docking experiments and point mutations, actually have been able to show the relevance of every amino acid, which we thought is actually part of the binding pocket. This is due to the fact that at that point, nobody ever managed to gain a co-crystal with an antagonist on the MR binding pocket. So we had to use this kind of model system, and here is shown, let's say, the result. As you can see, there's one expectation that actually shows that compared to the binding of spironolactone, which is shown in purple, the ketone moiety of the chromenone, which I already explained, is actually co-spotting with a ketone of spironolactone. But there's also a surprise, actually two surprises. The first surprise is that the dihydropyridine is really docking perpendicular towards the steroidal plane. And this was something which was astonishing at that time for us. Another important aspect is looking at the ester moiety, that the ketone part of the ester moiety is actually pointing to the C2 of the dihydropyridine. This is also a little bit counterintuitive, because you have seen, if you followed my explanation up to that point, that I always drew it in a different opposing conformation. And this was also intriguing to us. This we wanted to understand, and since this is a binding model, we actually tried to figure out more about it. So we were thinking about how to show that really the binding is, as I have to point it out. So we actually tried to incorporate this structural motif into an annulated ring system. And we decided to do that with a pyridone system. And here you can see that really, interestingly, the conformation, which is related to the ketone showing to the C2 on the right part, is significantly more active, than the one where the ketone is showing up more north, the oxygen. Okay, one aspect. The other aspect, we wanted to try out whether the ketone on the upper side is really already the optimum, or whether you can actually also use different kind of pharmacophoric elements to achieve a similar kind of effect. And in this regard, we were looking into literature, and we found other compounds that are working on steroid receptors, for example, bicatulamide or tanapogret, and these compounds have a nitrile that is pointing to the same kind of element that is actually in the backbone of this MR binding pocket. And here you can see that if you simply introduce the nitrile, the potency drops significantly. However, if you install an element at the ortho position of the head group, mimicking maybe a residual part of the formerly existing ring system, then you basically can keep the potency at a point that might be of use. So we wanted to bring actually these kind of two things together and actually match them. And this is shown on the next slide. So here, actually, you see when you marry these two elements, and this is actually leading now to a compound with 100 nanomolar, with actually having two good characteristics, also shown not only on potency and but also on the lipophilicity profile. So at that point, we made another fine-tuning and came up with a structural change that actually replaced the nitrile by a primary emit. And basically this led to an interesting compound which had a lipophilicity profile that was almost 1 log unit below the nitrile compared by log D 7.5. If you just install one methyl group, the whole potency drops and shows almost virtually no active compounding. And this is actually quite nicely understood if you look at our, let's say, binding model we could derive from the experiments we have been conducting. And there you can see that the primary amine is really creating an intramolecular H bonding network, and it's really also forming an H bond connections towards the serine 810, which would actually clash completely if you would install a methyl group instead of one hydrogen atom at the primary emit. If you're paying attention, you see that the compound on the right is not this compound I've shown in the middle. And this is actually basically due to the fact that it has another methyl group. And this is actually now leading to the last step of the optimization round, where we actually made a methyl kind of a screen to see what might be the best substitution pattern in this regard. And we have been able to show that with Finerenone, with a methyl group in the 8th position of the naphthyridine, it really has basically a high potency and virtually no selectivity problems towards any steroid receptors, and also with regard to the L-type calcium channel activity, which I now show a little bit later. So here, 2 or 3 words toward the synthesis. You see on the bottom right side that actually the hydroprotein synthesis is nothing very interesting. It's actually more than 130 years old. However, I would like to show that also some interesting synthetic concepts were also followed up on due to the synthesis of the compounds in question. I have shown you, for example, the amlodipine group. And this is, for example, using, shown in the green box, an hemiacetal starting point, where we actually hydrogenize the nitrogen group towards the aniline. The aniline can be condensed with an aldehyde, and then from the enamine reactivity can be cyclized with the open form of the hemiacetal, leading to the substitution pattern of interest. Then after a while, the acid needed to get converted by standard procedures towards the aldehyde, which was also working out fine. The sodium salt of the protonitrile shown on the left side can be condensed with the aldehyde I just mentioned, and in a 2-component cyclization, you really have been able to create, let's say, the hydropyridine of choice. The liberation release of the primary amino group was then conducted on standard conditions. Okay, this is one of my most favorite reactions overall. In the blue box, you see starting material for Claisen rearrangement. And basically, if you read it up to 200 degrees, nothing happens. If you go to 250 degrees, the reaction almost explodes because it's so exothermic that it's autocatalytic. So really, if you want to do that in a big apparatus, you really need to preheat the apparatus and really drop the the starting material drop by drop into the reaction funnel, and then the reaction is ready in 2 or 3 seconds afterwards. So afterwards, we actually isomerized the alkene and installed the diketo motif on top by a Claisen reaction. Cyclization was conducted under acidic conditions, and then Lemieux oxidation led to the aldehyde that then afterwards was used to undergo dihydropyridine formation. So Finerenone, this is a lab synthesis, was also synthesized from starting reactions materials. For example, the nitrile on the very left was originally vanillin that actually then was converted via Heck coupling to give the cinnamic esters, and then after Lemieux oxidation again, we could condense it with the beta keto ester as shown up front, on the bottom, no, on the top, I'm sorry. And this, after Claisen condensation, could be condensed to undergo the dihydropyridine motif. Then the alkylation was a little bit tricky. Whenever you use any reagent else than, for example, an Eschenmoser salt, then you were this was leading to a mixture of N and O alkylation. However, if you use the acidic conditions shown here with an orthoester, then the only product was basically the O-alkylated compound. Now, it's important to explain a little bit why I chose the two cyano ethyl moiety as on the ester. And this is due to the fact that you can mildly deprotect them by a retro-Michael reaction. If you do that, if you like to do that with an, for example, methyl or an alkyl ester on a dihydropyridine, this does not work. And this is due to the fact this is since these compounds are more like vinylic carbamates rather than simply esters. Then converting the acid towards the primary amide, and then the final racemic separation led to Finerenone. And this is actually still being done also on technical level with, let's say, a fancy method of column chromatography. But this is actually a different story that should be told by a process chemist. Okay, I have told you that Finerenone is at least as potent as spironolactone, and by far is more selective towards it. It's also more selective than on the L-type calcium channel than our starting points, and it did not show any deleterious effect on several enzymes and ion channels we have tested it. The pharmacokinetic profile looks, let's say, promising in rats, and this was actually our starting point for the pharmacological profiling. I do not want to go into too much of detail, but we tested this compound in a Doka salt model, which is actually a model for cardiovascular, oxidative, and inflammatory stress. And we have been able to show that if you compare the profile of Finerenone with eplerenone, you see that Finerenone is at least as efficacious at lower exposure levels, or no, those are doses, I'm sorry, as eplerenone. This is not, well, this is nicely matched on a biomarker level, showing here on proBNP, which is a clinically validated biomarker, and this shows impressively that the increase on efficacy is followed by a decrease on biomarker levels. Also on the morphological levels, we have been able to show that not only the heart weight is actually going down, showing the anti-hypertrophic efficacy on the heart as a target organ, but this is also accompanied by the similar profile on the kidney. So this means in the end that we not only have a drug that is maybe interesting enough to get followed up upon in terms of its heart failure potential, but also in terms of its kidney potential. And that actually led also to a more focused study on renal protection, shown here, where you can see, compared to placebo, that not only renal function is actually improved, and this is not actually the case to the same extent with eplerenone, but also on the markers on inflammatory and fibrotic enzymes and proteins. This is actually almost a perfect match. Okay. Looking at the properties of spironolactone, eplerenone, and finerenone, you see, although finerenone and spironolactone are equipotent, the compound, as depicted by the log D, is by far more hydrophilic finerenone. And this leads to completely different distribution pattern in terms of not only protein binding, but also vascular transport, tissue penetration, and distribution. So if you go and make an autoradiographic profile of finerenone, then you see a balanced distribution of finerenone, looking at the heart and also at the kidney. If you do that with eplerenone, you see much higher exposure levels on the kidney, 3 times. If you do that with spironolactone, this goes up to 6 times the levels you see in the heart. And this is illustrating also that these compounds are somehow different. Here I show some phase 2A results, and I have to walk you through the pick. Please start at the columns which are shown below the hyperkalemia or potassium level showing kind of diagram. And there you can see with 10 milligrams of the B94 8862, this was the former, let's say, code for finerenone, that it's at least as efficacious as spironolactone, showing on the right side. And if you then move up and look at the potassium levels these are associated with, you see that finerenone shows by far not the same kind of elevated levels of potassium, which is also quite promising in terms of, let's say, the therapeutic window of this compound. But most importantly, it's not about, let's say, the levels itself, but looking also at the adverse effects. You see that with spironolactone, you have always significantly more adverse events in terms of renal failure, renal impairment, but also, let's say, severe kind of hyperkalemia, as I already pointed out. In a phase 2B study, we actually decided not to follow up on the good, or not directly to follow up on the good data we have been seeing with cardiovascular or cardiac endpoints. We actually decided to go in patients with diabetes. And basically, the reason for that is that all the other steroidal antagonists, due to their strong effects on hypokalemia, they are not approved in these kind of patient collective. So we are able to compare towards placebo rather than actually to other compounds of this steroidal antagonist that could be considered as state of the art, which they are not in the diabetes collective. And as you can see, as we expect on the left side, based on the urine albumin creatinine ratio, you have and can see a dose-dependent decrease. And most importantly, no hypokalemia events are associated to efficacious doses as depicted on the right side. So overall, the study completely were considered to be successful. And this really triggered two studies. One is called the Figaro DKD study that is actually in patients with diabetes, looking at endpoints in which you are looking for cardiovascular events like cardiac readouts. Fidelio DKD is more related to the kidney performance in diabetic patients, and this study had actually for 2 months ago last patient last visit. So it basically already finalized, and now what our mathematicians are doing is they are doing they are cleaning up the database, and of course right now are in the process of preparing the statistics. So we hope that in the course of this year we will actually learn whether the endpoints are reached and whether the expectations with regard to the trials were met. So I told you now maybe a story of 15 years, and as I have mentioned, the pharmacology of MR antagonists is rather complex, especially if you have to figure that also the other steroidal receptors are somehow always at risk to be bound to. This is now maybe leading on a time scale to a point, where we hope that we can add something to the next level of innovation in this regard. Since also, let's say, to the start of our trials in phase 3, there was nothing actually out there that could create hope for these patients who are really at risk. And I have to say, well, since then, the SGLT2 compounds actually reached their endpoints, and this is also increasing the hope that these severe conditions, clinical conditions, can be treated well in the future. Since we really believe that MR antagonism as well as SGLT2 inhibition could actually benefit from each other. So overall, we really hope that Figaro and Fidelio, we have actually trials in place that really make a difference for the patients who are in need. So with this brings me to my end. I hope I did not, I'm a little bit 5 minutes over, so I have to apologize to everyone, and I'm very thankful for everyone who followed me throughout the last 50 minutes. So thank you.
Thank you, Lars. Sorry, I just muted myself. That was a fantastic talk. Amazing to be able to concentrate that much work into 50 minutes. We've got plenty of questions online. Before we go to those, can I just ask you a first question, which is, the complex pharmacology of the steroids seems to be due to their high log D, and your studies would just say it's just lack of specificity because they're distributing all over the body. So do you think you can transfer your dihydropyridine learning to other MR targets?
Yeah, it really looks like that. So what we have learned throughout the study, especially in terms of PK and PD, is really completely missing or actually hitting to the essentials of your questions. So it really looks like that you have to reach a certain level of hydrophilicity, really to get to a point that you can overcome, let's say, the complex pharmacology that is driven by the other steroidal receptors, which you do not want to deal with in this kind of patient collective. That seems to be true. Yeah.
Great. I'll hand you over now to Nessa and Fred, who have got lots of questions for you. If anybody else would like to ask a question, we always like to know where you're from. I forgot to say. So if you'd like to put on your question where you are in the world, it's always interesting to see.
Well, right now I'm actually in Brittany, but where I reside is actually in western part of Germany, close to Düsseldorf, in Wuppertal. That is actually where I work. But right now, I have to admit, I'm on my vacation, and I'm really lucky to have some good weather in western part of France. Yeah.
I think you're very lucky, Lars. Christine, first question.
All right. Well, we have one from oh, sorry to start with a big one, but this is kind of interesting. So from Colin McKinnon at Evotech in Oxfordshire, and he's asking about the chiral synthesis at process level. Where does the chirality come from?
Actually, the chirality is coming from the chiral chromatography. Yeah, we actually tried a lot to come up with a stereospecific synthesis, but although we managed somehow, it was always turned down because of commercial evaluation. So then the catalyst would have been so expensive, yeah, that you better can trash half of the compounds. And we actually found out, yeah, that if you're using a fancy kind of moving bed conditions for the chiral separation, this is actually the commercially most attractive case. So if somebody out there has a good, let's say, synthesis, chiral synthesis for these kind of compounds, that is maybe matching what we need to achieve in terms of, let's say, our commercial evaluation, then it's highly welcome. Yeah. What we do though is that the wrong enantiomer is actually getting oxidized and then getting reduced again because the oxidation is conducted at the dihydropyridine towards pyridine, and actually then the reduction leads to the formation of racemate again, which we then again can be put back into the process.
Okay, that's interesting. I was actually going to ask about whether there were any problems with the oxidation, because I guess I'm not that familiar with dihydropyridines, but they seem to be relatively easily oxidized.
Yes, this is one of the major problems. Yeah? And they are even getting oxidized in stereospecific manner. So under, let's say, physiological conditions, they form one compound with axial chirality, and actually not only one compound, one chiral form. Yeah.
We can carry on with this theme because we had a question in from Ewan Fordyce at the University of Dundee about the molecule itself existing as distereomers. can they exist as disterols, and did this impact the development of the compound?
I think this question is related towards maybe the conformational stability, rotational stability. And it looks like actually up to a temperature of more than 120 degrees, the conformation is stable. So means the head group is not turning around. It's more, it's really one enantiomer and not two rotational diastereoisomers or something like that.
We've got another
They asked if
Sorry, go ahead, Fred.
Okay, carry on. No, I was just going to say they asked the question about the impact though on the synthesis. Does it naturally form the correct isomer?
Whether we actually figured out what the, I'm not sure whether I really get the question. I'm sorry about that. Can you maybe point out the critical point?
Okay, I have to go back and look for it again. So the question was, if it could actually rotate, then obviously you would end up with a mixture. So when in the synthesis, did you end up with the correct diastereomer in effect?
Yes, must have.
It looks like, yes. So really, compound crushes out already on a rotary evaporator, and then you can simply separate it, and you do not get, let's say, more than you cannot really separate easily the correct and most potent, more potent enantiomer. It's not. I hope I do not make a mistake here, because this is already quite a while ago.
Okay, thank you.
Yeah.
Great. So we've got one from EK Ogba. Hopefully I'm saying that name correctly. I apologize if I'm not. So they're asking, is the selectivity and potency of the binding, is it more, I think you touched on this, is it more for the aldosterone receptor or more for the MR receptor as a whole? And I think they're coming at this from the point of what exactly is it that's preventing the androgen side effects?
I wish we would really understand. So we did not do binding model studies on the androgen receptor, I have to admit. Since we were not too much interested in this part, once we really managed to meet the challenge, on the MR. But this was basically, I think from a scientific point of view, this would be very interesting, but not, let's say, with regard to the task we have been facing.
We have an attendee from India. He doesn't give affiliation, and his name is Alminikan Bukunan, and he asked about incorporating peptide units, amino acids, in your studies, in your SAR studies. Did you ever try to incorporate such compounds?
We tried to, so actually I think we made at least something like 100 compounds that were supposed to be able to improve the solubility profile, because there's clear correlation between hydrophilicity and let's say metabolic stability in DHPs, which has been let's say published in a couple of publications, but at no end and no place in the molecule we have been able to find a place to incorporate these kind of things. And maybe there is also no good point or place where you can maybe reach something like point where you can move out of the binding pocket and reach maybe the environmental water or something like that, which would been great. But for us, it has not been possible.
Is that just a consequence of all nuclear receptor ligands?
I'm not totally sure because there are some nuclear hormone receptors which, for example, have as a pharmacophoric element carboxylic acids, like
multi-component reactions.
Yeah, but these are different receptors, and however, also looking at aldosterone and cortisol, they have some hydrophilic elements, but we did not manage to incorporate them into our molecules. This is basically, so we gave it up, basically. Yeah, whenever we tried something, this actually led to virtually inactive compounds. This is so. We tried to avoid this kind of negative learnings. And we were more concentrated on the things that were more dedicated towards the problem solving.
I had a general question, Lars, about the metabolites of these compounds. Did you have to do a lot of work on the metabolites as well to check them out?
Absolutely. And this was basically due to the fact that for us it was quite hard to believe how the compounds were behaving. So I've mentioned enantiomeric pure compounds were oxidized under physiological conditions to provide one enantiomer that is also chiral, based on axial chirality. And in this regard, we had really problems to believe that, but it was really shown in an extensive work that is also published by our colleagues from our developmental side, that this is really the case. We have 3 or 4 main metabolites, and in this regard, I think we have managed, of course, to identify all of them. But this was not so easy, I have to say. Yeah. But what the first thing that is always happening is oxidation of the dihydropyridine moiety, and it's not simple oxidation. So the metabolism takes place basically on the methyl group that is neighboring the nitrogen of the pyridine. And then it's eliminating to form, let's say, the pyridine ring. So it's not that it's, but this is something that we also had to figure out for our molecules.
I see we have a follow-up question on this subject of rotation or isomerizations. Yeah. Sven Nerdinger of Sandow in Austria comes back to this rotation. You said the rotation was activated at about 120 degrees C. He asked, was it possible there? Were you separating both diastereomers by using that sort of approach?
I think it is. Maybe there's a preference, to form maybe then the right orientation of the head group throughout the cyclization, but I'm not totally sure. This we did not really investigate deeper into that, I have to admit.
Okay, thank you.
Looking at the crystal structure, it really looks that the location of the head group is rather conserved.
I think that's it for questions, Caroline. I think the audience is satisfied with the.
It sounds like one. There's a very nice challenge thrown out by Lars for somebody to come up with a better stereochemical synthesis, and 2, there's a lot of interest in those rotational diastereoisomers, so it might be nice for somebody to do some work on that.
Well, I have to admit, these kind of topics were taken care of after we handed over this project to the developmental side. So still, when I handed over, there were more questions left than answered. And throughout, let's say, the last years, we learned so many things more about it. And as I have told you, if somebody has really a good solution for an enantiomeric synthesis for DHPs that are, let's say, attractive with regards to costs, I'm pretty sure a lot of companies would be interested in.
Well, thank you very much, Lars. It was a fantastic talk and really interesting to hear about Finerenone, which is clearly an exceptional compound. Very interesting to hear the story. So thank you everybody for listening and attending, and we hope we get to see you at some more of these talks. There'll be another one same time next week. Thanks everybody for listening.
Thanks also from my side.
Thank you, Theo.
Thanks very much.
Can you close the meeting?